Right. Okay, guys. So welcome to another episode of Med ABC, Acing Basic Concepts, right? So our topic for today is the iron profile, right? Now, what is the iron profile? Basically, what we're going to do, this is a diagnostic investigation. We're going to take a sample of the patient's blood, and we are going to look at the levels of iron as well as other iron-related markers that are present in the blood, right? So for us to understand the iron profile, guys, we need to first go through some basics with regards to iron metabolism in the body, right? So we will go through that first. So when we consider iron in our body, right, there is mainly a functional form of iron, a storage form, and a specific form of iron for transport purposes, right? So if we take a look at this, the functional forms, guys, it consists of about 80% of our total body iron, right? So you all know one of the major function is erythropoiesis, RBC production, right? Apart from that, we look at the storage form and the transport forms. They form another minute. The transport is actually the least, about 5% of our total body iron. Now, what I want you all to understand here, guys, iron never works alone, right? It tends to work in collaboration with certain other molecules. So, for example, in functional forms, you all have heard the terms hemoglobin, right? Myoglobin, and certain enzymes, right? Certain catalases, certain cytochromes. These are all parts. All of these molecules are in collaboration with iron, right? Then, if you take a look at storage, we are going to talk about something called ferritin. We're going to talk about another molecule called hemosiderin, right? All of these contain iron. And then the transport, guys, we're going to talk about a specific molecule called transferrin, right? We will talk about those in depth. The next point I want you all to know before we move further is when we consider iron, the absorption, transportation, and the storage, there are different forms of iron that are undergoing these processes. For example, guys, if we are taking iron into our body, basically via our meals, right, there are two primary forms of iron, right? The Fe2 plus and the Fe3 plus. What is this Fe2 plus? We call this the ferrous iron, right? That is the Fe3 plus. This is the ferric form. The Fe2 plus, guys, this red one here, right? The red one. The ferrous iron is what we also call heme iron, right? This heme iron is what we get from our animal sources, okay? Red meats specifically. Then when we talk about Fe3 plus, right? This blue one that I've drawn here, right? This is our ferric form. This is what we also call the inorganic form, guys. This is what you get from our plant products. Now, the Fe3 plus is also called the stable iron, right? So what I want you all to understand is, guys, for this absorption, transportation, and storage purposes, right? Our body prefers this iron in different forms. So for example, if we take the absorption process, that is absorbing the iron from the gut into this duodenal epithelial cells, right? It's going to take up this iron. For that purpose, our body prefers the Fe2 plus, is red, right? The heme iron, the ferrous iron form is preferred for the absorption process. Whereas for the transportation and the storage purposes, guys, our body prefers this blue form, the Fe3 plus, or the ferric form of iron, right? For the transportation and the storage purposes, right? So once we are clear with that, we can move ahead, right? So let me erase this, right? Okay, so let's start from the beginning, guys. Now let's imagine, right? There is some food coming in, right? And our GIT is going to start breaking them down, and eventually we are going to start absorbing. Now, iron is primarily absorbed in the duodenum, guys, right? So if we look at, I've taken a cross section here of the duodenum, and what we're going to do, y'all can see these small, tiny cells that I've drawn, right? These cells. So we are going to take one of these cells and enlarge it, right? That is this, right? Okay, we've taken one of these duodenal epithelial cells. Now, if we take a look at these duodenal epithelial cells, guys, what did we say? The first step of absorption, our body prefers this heme iron, or what we call the ferrous iron, the red one, right? Now, what is going to happen, guys? The ferrous iron forms can be directly taken up by the duodenal mucosal epithelial cells, right? So they will come here, okay? Whereas the ferric iron cannot be directly taken up. So what has to happen? This Fe3 plus needs to be converted to this Fe2 plus. For this purpose, guys, there is an enzyme here. There is a small enzyme here, right? I will draw it like that. So what is this going to do, guys? This is going to come, right? It's going to be converted, right? With the help of this enzyme, we are going to form Fe2 plus, right? Fe2 plus, and then that Fe2 plus can be taken up into the cell, right? So what is happening here, guys? 3 plus is being converted to 2 plus, right? So what is this? This is what we call a reduction reaction. So if it is a reduction, this enzyme is called, guys, perireductase, right? Perireductase. Now, this one is also called paraferritin, right? The names are not that important, but what I want you all to understand here is that for the absorption purpose, we need this Fe2 plus form, right? The ferrous form. Okay. Now, once absorbed, guys, two things can happen. One thing is this can directly go out of the cell and from there into the blood. We'll talk about that in a while. Another thing is, guys, depending on the body's requirement for iron, right? Another thing that can happen to this ferrous is that it can be stored. It can be stored within this duodenal mucosal epithelial cell itself. How does that happen? Now, if we take a look at this duodenal mucosal epithelial cell, guys, the nucleus has the genetic information to code for other proteins as well. One such protein, right, is this green one that I'm going to draw right here. This is a globular protein. We call it apoferritin. Right? What is the special thing about this guy? He takes up this Fe2 plus or the heme ion. He takes him up, right? And within him, right, inside, he converts this guy to Fe3 plus, the blue one. He converts it, this Fe3 plus, the ferric form, and then he stores it. Because if you all remember, we talked about at the start, for the storage also, our body prefers this Fe3 plus form, right? Fe3 plus. So this apoferritin molecule, this is called apoferritin. Once the apoferritin is loaded with Fe3 plus of ferric ions, we call it ferritin, right? We call it ferritin. Okay, ferritin. So apoferritin, right? Once it is loaded with Fe3 plus of ferric ions, we call it ferritin. Now, in reality, guys, I've drawn only one here, but in reality, inside a ferritin molecule, right, there can be up to 4,000 to 5,000 Fe3 plus ions, right? Okay. So that is the storage function. Now, again, what can happen with this stored form? Now, this is still in the duodenal epithelium itself. What can happen, guys, depending on the requirement? Sometimes this stored form, if it is needed, it can be taken up into the blood. If not, it will remain in the duodenal epithelium, right? In that cell, it will remain until that cell's lifespan ends, and it will slough off and go with our feces, right? So that is what will happen with this ferritin. Now, let's take a look at here again. We said some of the heme ion, it will directly come in, and from there, it can directly come to the blood. How does that happen? Here, guys, if we look at this step, so we have now the Fe2 plus or the ferrous ion, it's coming towards our blood. But what is the important fact we stated at the start? For our transport purposes, right? For the transport purposes of iron, again, our body does not like this red form. It likes the blue form, the ferric form, right? Okay, so for the storage purposes also, we like the ferric form, the blue form. For transport purposes also, we like the ferric form. So for that, again, there is another enzyme on the basolateral aspect of this cell, right? I will draw another enzyme here, right? Is another enzyme. This guy is going to convert this Fe2 plus, right, into Fe3 plus. Basically, he's going to convert the ferrous into ferric, and then that ferric ion can come into the blood. He can come into the blood. Okay, right? So this is the basic part about this absorption, guys, right? There are a couple of steps, well, with regards to converting the ferrous to the ferric and ferric to the ferrous, right? So I hope you all understood that. Then the next thing. Now, can we allow, right? So the ferric ion came right here. Can we allow this ferric ion to be transported by itself freely? No, guys. But this, our body doesn't prefer to send this guy alone because he will go react with other cells, right? Various oxidative reactions, right? So we don't like to send him alone. So for this purpose, our liver, the liver here, right? This guy produces some important proteins, guys. What are these proteins? We call them transferrin. Now I'm going to draw them like a W, right? A W shape, right? Okay, something like this. Okay, so you all also can draw it like a W. Now, why did I draw it like a W, guys? This is basically like a tiny sports car, right? It has two seats, what is going to happen? Each transferrin molecule, these are called transferrins, right? Transferrins. Each transferrin molecule can be loaded with two ferric ions. That means it will look something like this, right? Each transferrin molecule can be loaded with two ferric ions. Okay, right? So the important point here, guys, the liver, this guy is kind of excited to produce this transferrin. What do I mean by that? The liver always produces an excess of transferrin as opposed to what is required, right? Why does the liver always produce an excess of transferrin? So that whatever ion comes from the GI tract can be absorbed, right? And transported. Okay. So for example, guys, what do I mean by this? For every 3 transferrin molecules, now I've drawn 3, 1, 2, and 3. For every 3 transferrin molecules, only 1 will be saturated with ferric ions, right? Only 1 will be full of ferric ions, right? The remainder will be empty. So if I are talking in terms of saturation, right? If we talk in terms of percentage saturation, right? The percentage saturation of transferrin, guys, is usually, what is it? 1 in 3, right? One third. What is this? That is about 33%. Now, in your books, guys, you will see the range given. Usually it's about 25 to 50%, right? 25 to 50%. So we'll take it as 33% for the sake of today's class, right? Okay, so what does this 33% or in your books 20 to 50% tell you? In a normal person, about 20 to 50% of their transferrin, right, is loaded with ferric ions, right? Right. Then, just to recap a little, guys, what did we say now? There is a molecule called apoferritin. This is a tiny recap before we move on forward. There is a molecule called apoferritin. That apoferritin, once it is loaded with heme ion, it will convert that to ferric ion, right? And those two together, right, will form the ferritin, right? So you all can draw it something like this. Okay. Right. Then the next point we talked about, there is transferrin. We talked about transferrin, this W-shaped one. And we said each transferrin can be loaded with 2 ferric ions, right? So again, if I draw it like that, right? 2 ferric ions. And here, guys, what I want you all to know, the important term, once a transferrin is loaded, fully loaded with ion, we call this total molecule, we call it serum ion. So what is serum ion? Transferrin loaded with ferric ions, right? That is your serum ion. Okay, right? Okay, so let's move on to the next thing, right? So once you've understood this, guys, what I want you all to know, in our blood, there are free transferrin molecules as well as serum ion, or what we call bound transferrin molecules, right? Bound, they're already saturated with ion. Ion has gone and bound to them, right? So if we take a look at this free and the saturated, so we have free transferrin plus the saturated transferrin, saturated with ion, saturated transferrin, right? These two together, guys, we call it as the TIBC, right? Total ion binding capacity, right? So what does this indicate, guys? This indicates how much of ion can be bound, right, to the transferrin in our body. I will give a better example in a little bit, right? Okay. So once you've understood this, right, next we'll go to the storage function, guys, right? Now we talked about absorption and we talked about transportation, right? So we'll come to the storage function. When we check the storage function, guys, okay, look at this. Let me draw here, right? I've already drawn it. There is a red blood cell. Now you all know one of the primary functions of iron is erythropoiesis. So you all know iron will be loaded during RBC production. Iron will be loaded right into the RBC, and then this RBC will live for about 120 days, guys, right? This is the life cycle. After 120 days, right, this RBC needs to be broken down. So for that purpose, the RBC will come to the spleen. In the spleen, guys, there are special cells, some special macrophages. We call these macrophages Kupffer cells, right? Kupffer cells. Okay, Kupffer cells. So what's going to happen? This RBC, once its 120-day lifespan is over, it's going to come here and it will be broken down for recycling. Here, guys, the recycling process is a little bit large. We're not going to go into detail of that. But what I basically want you all to know, right, it will be broken down, right, into one, the cellular component, right, the cellular component, and the other one, right, the hemoglobin. The hemoglobin will also be broken down. The cellular component we are not going to discuss, guys. It will be broken down into its constituent molecules, amino acids, fatty acids, phospholipids, right? All of that, right? Hemoglobin, how is it broken down? So it's broken down into the heme part, and the globin part, right? Heme part and the globin part. Now the globin is the protein, right? So that will be broken down into amino acids. We are not bothered about that. The heme part is broken down into something we call biliverdin, guys. Biliverdin, okay? Biliverdin, and together with this biliverdin, I will denote it with the letter B. This is biliverdin, right? Biliverdin plus we get our heme iron. Our heme iron, remember, in red, right? In red. This is, so the hemoglobin is broken down into heme and this biliverdin, okay? Right? Now, the biliverdin, it will go to the liver where it will be recycled, right? It will be excreted and it will be recycled, right? The red, the heme part, right? The heme iron, or what we call the ferrous iron. What is going to happen to this guy? Look at this, guys. Now, just like our duodenal mucosal epithelial cell, our Kupffer cell also has a nucleus, right? Also has a nucleus. And this nucleus codes for certain proteins, right? Certain proteins. One such protein, again, just like our duodenal mucosal epithelial cell, is this protein we drew before, our apoferritin. Now, the same process that happened here is going to happen here as well, guys. Some of this apoferritin, they will take up this Fe2+, right? And they will convert it within themselves. There are some enzymes, right? They will convert it to this ferric form, the blue form, and it will store these. So I will draw a couple just for us to understand, right? So I will draw several. Once the apoferritin is loaded or is having this ferric ions, what do we call it? We call it ferritin, right? Ferritin, right? Now, when there is a lot of ferritin being produced, guys, what is the important thing I want you all to know? Some ferritin, if it is produced a lot, it can just leak into the blood. It can just leak into the blood. Okay, right? Something like this. It can leak into the blood. Okay. Now, what is the importance of this, guys? What is the importance of this? When there is a lot of ferritin being produced, right? Some of it can leak into the blood. What does this tell you, right, about this ferritin that comes into the blood? We call this ferritin that comes into the blood, we call it serum ferritin. Serum ferritin, right? So, what does the serum ferritin tell us, guys? Our serum ferritin is a direct indicator of our stored iron. Because we talked about stored iron, we said ferritin is one form of stored iron. So when ferritin comes into the blood, that becomes, we call it serum ferritin. And serum ferritin is a direct indicator of our iron storages. So what does that tell you? Serum ferritin, guys, we say it is directly proportional to our body's iron stores, right? Body's iron stores. Now, what's the importance of this, guys? If we want to check a patient's body's iron stores, we don't need to cut into the bones. We don't need to drill into the bones and take a sample and then look for iron. No, we just take a blood sample and we check the serum ferritin because that directly indicates the body's iron stores, right? Okay. So this is one thing that happens when there is a lot of ferritin being produced, right? They just fall into the blood. Okay. They just leak into the blood. Another thing that happens here is, guys, these ferritins, they can form aggregates. Now I will erase this a little just to get some room, right? They can form aggregates like this, right? They can form aggregates like this. Okay. Now this is the same ferritin, right? Yes, with some enzymatic reactions going on, right? They can form these aggregates. Now, what are these large aggregates of ferritin called? We call this hemosiderin. Hemosiderin is a large molecule, guys. It's a large molecule and it is an insoluble molecule, right? Now, this is also called our stainable form of iron, guys, because this hemosiderin is so large, you can stain it. There is a stain called Prussian blue, right? We can use this stain and we can actually observe it under the light microscope. Okay. So that's how large it is. Now, this hemosiderin, like the ferritin, it does not, unlike the ferritin, right? It does not just leak into the blood, right? This guy is insoluble. He will stay there, right? He will not leak out like that. But what the important thing I want you all to understand here, right? This hemosiderin is not a permanent molecule. It is temporary. It can, whenever needed, right? It can again be broken down into these individual ferritin molecules. It can be recycled. And whenever needed, the ferritin will be brought into the blood as needed. Okay. So that is how our iron is stored and taken into the blood whenever needed, right? So now you all are familiar with the term serum ferritin as well. The important thing I want you all to understand about the serum ferritin, guys, serum ferritin, once it comes into the blood, right, it has the ability, right, for this to disintegrate. What do I mean by disintegrate, right? This covering, this outer covering will be broken down, right? And then the iron, the ferrics that were inside will be released. We said each ferritin molecule can hold 4,000 to 5,000 ferric ions, right? So these ferric ions will be released in the blood, and immediately, who will take up the ferric ions? Our transferrin, right? So our transferrin will be here, right? And these guys will take the ferric ions, 2 ferric ions per transferrin molecule, right? And they will be transported to the required destination. Okay. So this is the basic concept I want you all to understand, guys. Then let's talk a little bit more about our total ion binding capacity, just to make it clearer for you all, right? So if I erase this portion, right? Let me erase this portion, right? Look at this, guys. Now, why are we doing the iron profile? Usually we are doing an iron profile in a suspected case when a patient comes to us if he's showing clinical features of iron deficiency. This is one of our primary reasons, right? So let's imagine that we have a patient in front of us, right, who is now iron deficient, right? We have an iron deficient patient in front of us, right? Iron deficient. Now, if he is iron deficient, right? If he is iron deficient, what is going to happen, guys? There is less amount of iron in his body. So whatever his stored iron, now we talked about how iron is stored, specifically in Kupffer cells, right? As ferritin, iron is going to be stored. Whatever he has stored will be quickly released into the blood, right? It will be quickly released into the blood so that his body can utilize. Now he's iron deficient, right? So his stores will quickly release these stored forms of iron, right? Specifically the ferritin, right? Once the stored iron comes into the blood, guys, what will happen? Since he's iron deficient, it will quickly be utilized, right? The serum ferritin that arrives here will quickly be used up, right? So if you take, now he's an iron deficient patient, if you take a blood sample and you check his serum ferritin levels, it will be low because it's being used up, right? Serum ferritin is being used up, so it will be low. Now, if his serum ferritin is low, what does that tell us about the iron stores? The iron stores are depleted. Iron stores have been used up because we said serum ferritin is a direct marker of the body's iron stores, right? So the iron is being used up, right? So one thing is that the body's iron stores are reduced, depleted. The other thing, when we talk about serum ferritin being low, whatever iron, we said this serum ferritin will be disassembled and the iron will be released into the blood and then will be taken up, right, by the transferrin. If the serum ferritin is low, this iron is also going to be low, right? Okay. So what is the other term, guys? We said for this transferrin that has been loaded with iron is also going to be low, right? What is this? We call this a different term. We called it serum iron. So serum iron is also now going to be low in this iron deficient patient, right? I hope you all understood up to this point, right? Now, when serum iron is low, what is our liver doing? This guy is always enthusiastic, right? What is he going to do, guys? He is going to start excessively producing a lot of transferrins, right? He's going to produce a lot of transferrins. How is this going to look, guys? Look at this. Let's break this down into 3 easy. If we take a look at a healthy person, right? If we take a look at a healthy person, right? I'll put healthy percentage. Healthy, right? If we take a look at a healthy person, right? I will draw some transference. I will draw transference like coalesce together, so that it's easier, like this, right? This is our diagram. So if we take 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, okay, 10, fine. If we take 9, you all know 3 will be saturated in a healthy person because we said it's 1/3. It's 1/3. If we take 9, about 3 will be loaded. 3 will be loaded with iron, right? You all understand this, okay? Now what is happening, since the liver is hyperactive in this iron deficient patient, liver is going to go into hyperactivity and it's going to start producing a lot of this transferrin, right? So when there is a lot of transferrin produced, guys, what is it going to look like? Something like this, a lot of transferrin, right? Okay. However, this patient is iron deficient. Now later in another chapter of iron deficiency anemia, we'll talk about the causes of iron deficiency. What does that tell us? There is either a less amount for iron deficiency, guys. There's multiple reasons, right? One is reduced intake, right? Another one is reduced absorption due to some reason. Another thing is increased losses, right? We can talk about worm infestations, right? There are increased losses, right? Or even conditions like menorrhagia in females, right? There are increased losses. And another one is increased demand. So whatever those causes are, in an iron deficient state, there is no iron to fill all of these transferrins, right? Transferrins are being produced in excess in the iron deficient state, but there is no iron, right? Let's say there is only like 1 being filled, right? Now imagine this like a classroom, guys. Imagine there's a classroom. There are 50 chairs in the classroom for students to sit, right? In this classroom, let's take one example. On one fine day, right? There are 40 students who have come. 40 students who have come in. So there's 10 seats remaining, right? 10 seats remaining. So there is a capacity remaining of 10. Whereas let's take another day, right? Like a Friday evening when we all like to, cut class a little, right? On a Friday evening, in that same 50-seated class, there is only, let's say 5 students who have come in, right? What is the remaining capacity? The capacity is going to be 45. 45. This is the same principle like our total iron binding capacity, guys, TIBC, right? In an iron deficient patient, this is the same thing we are going to see. When there is iron deficiency, there is going to be an excess, right, of transferrin being produced, but there is no iron to bind. So the remaining capacity is very high, very high. So the total iron binding capacity increases, right? So just to summarize what we've learned. So we've covered everything regarding the iron profile. Just to summarize, guys, the 4 most important terms I want you all to know in your iron profile, right? One is the serum ferritin. Write it in red, serum ferritin. What was the serum ferritin? Just to recall again, this was the ferritin that leaked into the blood, right? Specifically from the storage, like the Kupffer cells, right? Okay, serum ferritin. Another one is our serum iron. Serum iron. What was our serum iron, guys? This was our ferric ions. Once they will be loaded into the transferrin, we call it serum iron, right? The other thing is our total iron binding capacity. Now this we've discussed already, TIBC. And the last one is our percentage saturation. What is this percentage saturation, guys? How much, right? How much of the transferrin has been saturated with iron, right? So if we take an iron deficient patient, right? A patient with IDA or iron deficiency anemia, right? If we look at the serum ferritin, right, you will see it reduced. Now you all understand why, right? Because the stores are being depleted, right? The next thing, if you look at the serum iron will again be reduced, right? Okay, you all can now understand. The TIBC, what happens? TIBC, the total iron binding capacity, due to the overproduction of transferrin by the liver, the TIBC is going to increase in iron deficient patients, right? And if you look at the percentage saturation, so what do I mean by percentage saturation? There was so much of transferrin, right? But only one was loaded. One was loaded with iron, ferric iron. So the percentage saturation is going to be low, guys. Okay. This is our basic iron profile. Now we will talk about in upcoming chapters how to apply these same concepts, right, into other disorders. So our next lesson is going to be about thalassemia, right? So we will discuss further in detail about this iron profile, right? So I hope you all understood today's lesson, guys, right? So that's about it for today's lesson. drop a like, subscribe to the channel, and catch you guys in the next one.